PhD in chemical engineering from University of Connecticut. After that, he did a postdoctoral training in Cornell University. Next, he joined Syracuse University in 2006. Currently, he is Stevenson Endowed Professor in the Department of Biomedical and Chemical Engineering and Associate Dean for Research in the College of Engineering and Computer Science. He has received many awards, including Early Career Translational Research Award in Biomedical Engineering from the Wallace H. Counter Foundation and NSF Career Award in 2011. He was named the College Technology Educator of the Year by the Technology Alliance of Central New York in 2010. He is also a recipient of the Faculty Excellence Award from the School of Engineering and Computer Science at Syracuse University in 2014, Chancellor's Citation for Faculty Excellence and Scholarly Distinction in 2018. He has 72 papers with more than 7,000 citations and an H-index of 40 and 13 issued pending patents. He has broad research interests in microbial control and the safety of medical devices. His research has been funded by NIH, NSF, EPA, DOE, Coulter Foundation, Sloan Foundation, and industrial sponsors. He's a fellow of American Institute for Biomedical and Biological Engineering. The floor is yours, sir.
All right, thank you so much, Mohammed. It's a great pleasure to be here. So I want to show some of my work. I'll start by showing this opening slide, which is a bird view of our campus in upstate New York. If I have not been here before, this is a beautiful place, just about, I would say, 240 miles north of New York City. If you use a metric system, it's about 400 kilometers. We're actually closer to Canada than New York City. This is the fall view of the campus, and the campus is going to be like this in just a couple of weeks. And after that, we'll see this change, and towards the winter, we don't talk about that. So this, using my pointer here, I don't know if you can see that. So I'm pointing to this building. That's the home of our lab. My own lab is at the corner of this building. We do have a nice view of the campus. So my research as engineer, I'm motivated to solve some problems. And the problem we're interested in is antibiotic resistance. As you might have seen many news about the development of this resistance problem, essentially we're seeing some of alarming cases that we call superbugs that are resistant to all antibiotics we have. And that presents a major issue that WHO predicted that if we don't control that, this will become a major issue by 2050. It can cause more than 50 million deaths per year. Last year alone was already 2 million globally. And if we don't find enough new antibiotics to solve this problem, and the future is gonna be an alarm to us. So we need to do something different. And this one I show you here is an example of how it began. This is rather old data. Actually, ever since we had discovered penicillin back in 1928, scientists have seen resistance. Essentially, you take antibiotic, which is supposed to kill bacteria, but it doesn't. And in that case, we cause resistant strain, as we know. This one shows in 1980s, this is of Staph aureus, which is the one that causes sore throat and some other infections. And the typical antibiotic used to treat that is called methicillin. If you take the strain and you treat it with the methicillin, you don't kill it, we call this methicillin resistant Staph aureus, or MRSA. And you can see as I'm pointing to this, is increasing almost exponentially over time. At the same time, we're drying the pipeline of new antibiotics. This data shows up to 2012. After that, there were a few new antibiotics being approved by FDA, but they're all derivatives of the ones we have seen problem with. So this together shows us an important call that we can think about what can be done differently and what can be both like strategically and also to understand the mechanism. So essentially, potentially a paradigm shift in the way that we fight infectious diseases. I think this problem is rooted in the way that we did antibiotic discovery. So this is the flow chart of how it works. Basically, we have this, you can see, you take this culture medium of something to test the bacteria that you're interested in testing. You put a different amount of antibiotics in those cultures. You increase the concentration and you or to form this gradient, and you test it. Overnight, you will see bacteria will be incubated. On the second day, you want to come to check what concentrations allow the bacteria to grow and what don't. And the lowest concentration that stop the growth altogether would be called minimum inhibitory concentration, or MIC. And if the MIC is low, that suggests the drug is very effective. Then potentially, if you further test cytotoxicity, you may find a winner that can go down the road until approve the drug to be a new antibiotic. If the MIC level is high, that will indicate this drug potentially can be toxic because you take a lot of drugs to kill this pathogen. So by using that, you can find the possible leads and move on. But this important test, which led to the discovery of all antibiotics essentially we have in clinic today, is based on several assumptions. Number one, we need the cells to be able to grow, because you use the growth to test if there is or there's no inhibition, right? And then we also need pure cultures in most of the tests to get control of the cultures can test this in a reliable way. And if you do have multiple species in there, it's possible to do, but if you don't control the balance well, it becomes challenging to study. So in many cases, people use pure culture to screen this. In real conditions, this may not be the case, right? We also assume homogeneous cultures. The cells are well mixed in the medium, so you can test this reliably. So these three assumptions are important for this test to work properly, but are they the case in real world for the infections? Probably not. Even in the lab conditions, if you put bacteria on the surface of an agar plate, they will not form a homogeneous population. On the left, we show here is this is almost like a flower. Those happened to be my first experiment when I did as a PhD student years ago. At that time, I didn't even know enough about bacteria, just put some on the agar plate. Second day, I saw this gigantic, actually quite beautiful structure to my eyes, at least. This is about 4 inches across, was pretty impressive. I thought I discovered something new, but the literature search told me this is not new. This has been known. It's called swarming. So what is swarming? What is swarming? Swarming is referred to a group of cells or other living creatures move together. As we know, the bees swarm in the springtime. If the nest has 2 queens, one of them has to leave. When she leaves, she will bring a lot of working bees with her, and they move together. It's called swarming. So bacteria do exactly something like that. If you zoom in under the microscope at this one of these fingertips, you can see the bacteria form in bundles that they move together. So by doing this, cells also become longer, and then therefore gain some resistance to, for example, host immune cells. If you have phagocytosis, it's very hard to engulf them by macrophages or neutrophils. The bacteria also become stronger against other stress conditions. It turns out they also use similar activities to move along biomaterials to cause infections, such associated with catheters. On the right-hand side is so-called biofilms. You can see these dome-shaped structures, and they're formed by multiple layers of bacteria. Actually aggregated together, they build this kind of complex structures. Some of them even have water channels between the clusters for removal of the waste and transport of nutrients. So they definitely show they're not homogeneous culture. That is not the same as the MIC test is based on. But how common are these in infections? In fact, they're pretty common. So just a few examples. Medical device associated infections. This is involved in almost any implant you can put in human body, or even partially indwelling devices such as catheters, urine catheters. They all can have biofilm infections. They are commonly found in cystic fibrosis-related lung infections. Many hospital-acquired infections involve these biofilms. UTI already mentioned. The chronic wounds also have a major problem with biofilms. All of this, you can see, they're not the typical acute infections that you take antibiotic, you recover in a couple of days. These infections last, and they may last for days or weeks, months, sometimes maybe even years. So this is what you have in the case of infection. We take antibiotics. I think I showed the time as hours, probably longer. This hours will be in vitro In human bodies, it could be days or sometimes a little longer. But if you do eradicate this, you'll be okay. In most acute infection, we have experienced that are like that. If you have a resistant strain such as superbugs, they will not be eradicated, so the infection will remain. That will require change in the therapy. But you see in clinic, there are other type of this called the tolerance, which is more of the ones that show on the left. If you take the strain out of that patient, you test them on the bench, they're highly sensitive, just like rather normal cells. But in the human body, they behave just like resistant infections. So this is called tolerance. So tolerance comes from the disease symptom, not the genetic makeup of that infectious bacteria itself. So there's something happened to the cell to become tolerant. It has been shown the biofilm is part of that. And in recent years, it show there's another behavior which is more related to tolerance, or basically in this more extreme case of tolerance is like a dormancy. So these are called persister cells. These cells, for some reason, they go to some phenotypic changes. They're not genetic, but they become highly tolerant to the stress. Now, they're not the whole population either, so they're part of this, usually a small population. So what you see in those cases is at the beginning of the treatment, you can see you have this rather normal killing to begin with. After that, you reach this plateau. Even you extend this for a longer time, it doesn't get eradicated. So if you stop the treatment, they will come back, reestablish. Some of these dormant cells will go back to wake up again. You restart the cycle. So the infection continues. With both of them, the tolerance coming from this biofilm itself, and also the biofilm that harbors the persister cells contribute to this long-lasting chronic infections. Eventually, bacteria can have a chance to mutate and become permanently resistant strains. So this cartoon here shows the multiple steps of this biofilm formation. Essentially, there's a first interaction with the surface itself, and then the cells will, if they like the surface, do produce polysaccharides and some other matrix materials such as proteins and extracellular DNA as well to form this showing purple color as a matrix. At some point, they can break the structure and swim somewhere else. So this is in the research community of this, we also call this kind of microbial version of cancer because they're both hard to get rid of due to drug resistance, and also they can move and expand. In cancers, we call metastasis. In biofilm, we call this dispersion and the reattachment. So this whole cycle continues. Now, my research group is basically motivated about this problem associated with biofilms and tolerance. So we're trying to make some contribution to the field by studying, on one side, we study biomaterials because the materials are important as a hotbed for this infection to begin with for the medical devices. We want to learn how bacteria interact with the materials, and then can we learn something from that to get some rather rational design to prevent biofilms or remove biofilms. On the other side, we're trying to understand why bacteria become dormant or form those persister cells. Then can we have some new ways to treat those dormant cells? So I'll start by talking the biomaterials part and use just a few minutes on the drug discovery side. In terms of the biofilms, when it's biomaterials related, they can be on any biomaterials like put in the human body. There are some examples here. Actually, see there are millions and millions of devices put in a human body every year, ranging from soft to hard tissue contacting devices. They could be polymeric, they could be metallic, or maybe a combination of them. And some smarter devices now have power units in there. Devices get more complicated, some complicated structures as well. On the surface, they can provide hotspot for bacterial attachment and cause complications, and infection is certainly one of them. Now, what's interesting about this is if you think about the medical device-related infections, they happen in the same location of just normal human tissues, right? So any part that you need a device to fix certain things about healing or restoring a function of the human body, that location could have potentially an infection due to that device. But you also can have normal infection for the same organ as well. But there are some big differences between them in the way of how hard it is to get rid of them. Because they involve biofilms, all of those infections, when it has a medical device involved, they become hard to to control, to get rid of, right? There's high level tolerance to antimicrobials we already talked about. And then we don't have effective treatment. On top of that, it's interesting to know that when you have a biomaterials implanted, the minimum number of bacteria cells that require to cause infection or cause the infection load is reduced by 1,000 times compared to normal tissue. So the bar is a lot lower. You need much fewer cells to cause infection. That causes a problem too, right? But if they do colonize and become biofilms, they become up to 1,000 times more tolerant to antibiotics. So we can see it's almost like 1 million times difference in terms of the starting of the infection and eradication of the infection. That causes major problems. On the therapeutic side, basically it's impractical to use antibiotic to get rid of the infection because it's such high dose that patient couldn't tolerate. Now, but why is this? We believe this is rooted in the way that we engineer the biomaterials because they are all abiotic materials. They don't have a biological origin, and they're human-made materials, so they don't have the innate immunity. Now, if you do put in human body, the immunity also doesn't work as effectively, which I will show in a second, when you have bacteria colonizing those artificial surfaces. So conceptually, the best way to control this then is to get rid of the biofilm, so don't let them attach on this. So if you can prevent bacterial attachment from the very beginning, then we don't have any problem about this, right? That's the idea. But this is such a non-new idea. People have been doing this literally for a long, long time. Even back in the 3rd century, Greek people knew if you put wax on the sheep hull, they can move better in the water and get less falling from the microbes and algae in the water. That's biofilm too. So since then, though, we need new technology to do this better. Of course, in the last 2-3 decades, we witnessed a lot of exciting research done in the field, either by chemical, physical, or biological modification of surfaces that you can reduce microbial colonization. Essentially, you can create non-falling surfaces. However, when you move this to in vivo testing and try to move forward, we still see a major challenge about long-term biofilm control, in which we need to inhibit initial biofilm formation, but also because regardless what we have right now, nothing can permanently prevent everything forever. So once bacteria come to this, they will overcome the features we engineer. So we need some solution to control and eradicate the mature biofilm as well. We need to do both, and that's not the easy task. So part of the research in our lab is try to engineer biomaterials and surfaces to have some sort of self-defense mechanism, maybe inspired by human tissue, for example. And if we can do this right in the long run, we hope to engineer smart anti-fouling materials. We hope they can be species non-specific, so they don't work with just one bacteria, but actually multiple species. We hope them can be programmable. And so you can, for example, if a force to be generated or release of the antimicrobial, they can be programmed and they become responsive to microbial challenge rather than passively released or static control. And then hopefully we can control biofilm formation without using antibiotics, or at least have a synergy with conventional antibiotics, because we know there's a problem using those in the long run due to resistance problems. So with those in mind, we look out to see how can we control this. One way we wanted to think conceptually is maybe we can think about how bacteria make a decision to form a biofilm. That's what we call how bacteria read the map. Essentially, this is how we do, right? I show the map of United States here to show human activities. Activities have to look at night, not during the day, because the lighting shows activities. You can see there's a major metropolitan areas, LA areas, for example, right? New York City, we're in Syracuse, we're here in this small dot here, smaller city, but there are also areas that we don't have any people living there. So we use our information as we examine the map to decide where to go. But do bacteria do something like this too? In fact, I think they do. This is one picture on the right showing E. coli biofilm taken with fluorescence microscopy. And the green color was done with live dye staining. So many of you know this. Green means live, red means dead. This is a healthy biofilm. There's no dead cell. But you can see though, the cell clusters, they're not uniform on the surface. They're big clusters, smaller clusters, and some open areas that they don't have cells. Almost just like our map. We may have a desert that nobody wants to go there. There may be something called bio-inert area, or for other reasons, the bacteria don't want to go there. So do they sense this just like we do for our map reading? Now, the difference though is we have eyes, we have ears, we have nose, we have brain. We can process information to do prediction and planning. Bacteria don't have those, but they do have sensitive sensing mechanisms about chemicals, about physical features. What that means is basically touching. They can use touch to sense. Probably Andrew appreciate it. But what do they sense in terms of touching? They can sense those chemical and the physical features, right? Specifically, I want to emphasize the topography, which is a focus of our work. So when it comes to talk about the topography, essentially you can think about the medical implant once you put in human body with some sort of topographic features. This is essentially a battleground. There you have immune cells, try to colonize on this to clean any possible microbes, and microbes try to hide somewhere to survive. And then eventually, if they win, they will build this like biofilm shown here. If the immune cells can get here, get rid of them, you don't have infection to develop. And then if this is tissue integrating surface, then we hope the tissue cells will get in and form this integration before bacteria can come. Then the infection rate is dramatically reduced. Right? So this is the representation of this interaction of the many different cell types on the surface, which still don't have a very deep understanding, although there's certain things we have learned from this. I'm going to show you a real example to show what I mean here. On the top, you can see this is a macrophage, and the yellow ones are macrophages. The green ones are E. coli cells. They are put in a co-culture model on a piece of silicone, which is PDMS polymer. And then we created those recessive features. There are hosts that went into this. You can see bacteria can go in the host. Macrophage will come over. What we found interesting is if you have a small hole, bacteria can easily get in. The phage may not entirely squeeze in. They can try to reach down. So if you look at the lower one on the left, this phage reach into this wall, engulf this E. coli bacterial cell inside this green one. So this phagocytosis is successful. But if the biofilm established first in this on the one on the right, you can see the macrophages is not able to clean it up. So this kind of interactions happens on many medical device surfaces. The best example known probably is the breast implant, as there was a recall in 2019 due to very similar features like this and caused bacteria to colonize. And through some still not very well-known mechanisms, they cause immune response and trigger the formation or development of a type of lymphoma. It's not breast cancer, but it is immune system-related cancer, and that's cause fatal cases. So there's a lot of questions to be answered, and we can learn from this. What kind of features should be avoided? What might be actually better to help solve this problem? So this is an example of bad topography. Then I'm going to show you a couple maybe called good topographies. I want to go back a little bit to some of my early work. That's how we got here. So this back to 2011. This is 12 years ago when I was still assistant professor at that time. I was trying to get a quick paper. I talked to actually my first PhD student was also the best at that time. I said, can we make this kind of protruding features on the surface? If we can make this and put the bacteria down, we want to see where they go. Potentially, they don't want to go to the top. Maybe they want to be between this. But if we separate this further apart, things may change. So we want to just study this. Indeed, what we saw was we got this very dense red color showing the E. coli cells labeled with red fluorescence protein expressing gene. They colonize the space between those pillars. But when we try to quantify this biomass to get a figure showing on the right lower corner, we quantify the biofilms on top of the pillars as well. And when we plot that one, we saw something quite unexpected. Essentially, we saw this threshold kind of behavior. When the surface area was pretty small, there was essentially no biofilm on it. When we have this large area, then we'll see much bigger, much more biomass on those. But we didn't understand why bacteria will do this to form this kind of threshold. We really had no clue about it, but we have enough data to report what we saw at that time. And then, but we wonder what it might be. And 2 years later, we had a high school student very interested in research, and we talk about possible science fair project. We thought maybe we can do we call it bacterial balance beam. So if we make this line patterns using topography, we can make cells line head to toe, kind of form a line, and that will cause this interesting alignment. That's our hypothesis. But the results wasn't quite as we expected. In fact, we found more cells were aligning more like a perpendicularly to this orientation of the line, such as this cell shown here. To make conclusion, though, we have to count them, make calculation, and run statistics. As we did so, we call them perpendicular if they are within 30 degree in the perpendicular orientation to the orientation of the line, or diagonal or parallel. When we count the cells, many of them, essentially we found this trend. So when the pattern is narrow as 5 micron across, we saw the majority of them, well, half of them, I would say, wanted to align more in the perpendicular orientation. And then fewer cells, about half of them, compared to this in each category, to be parallel or diagonal. And when the pattern gets wider, this preference basically or bias disappears. On the flat surface, it became purely random. We were actually more confused about this, and just like the other threshold phenomenon, we couldn't figure out why this is exactly the case. But what's interesting, and later when we did some SEM picture with my talented student at that time, later became postdoc, she found that the flagella may be the reason, because we saw the flagella was involved in the attachment here. So, but how would that affect this? We think actually if the flagella is involved in this kind of sensing of the surface, might be that will indicate some kind of preference that led to this preference. So is that the case? So what we did was we follow the cell after they attach on the surface. You can see here, this one is cell. This cell attaching on the flat surface. You can see cell body rotates. And the size doesn't change much. So that means the projection didn't change much. The size or the angle of the cell doesn't change much. You can see a whole circle, right? But it remains similar angle there. But when the bacteria was attached on the side of the pattern, you can see the size seemed to change quite a bit. It's not the cells are getting longer or shorter. It's just the angle was changing. So it's all over the place. So when we track the cells, you can see if the cells are tethered on a flat surface or a rather wider pattern, you can see this kind of circular motion of the cell. When it's tethered on the side of this, they form this rather change the pattern, and the cell angle changes a lot as well. So this tells us two things, actually. If the flagella is involved, then it explains why we saw more cells aligned perpendicular on the narrow pattern, because the pattern we tested was 5 micron tall. So you get 5 micron on each side, and across is also 5. So we have twice more area on the side to attach than the top. But if the cell does attach on the side of it, the only way they can land on the surface will be going across the surface, which will be a perpendicular orientation. So as the pattern gets wider, there will be less area on the side compared to the top, and then this kind of bias will disappear. That's exactly what we observed. He also tells us if this cell body, the angle changes significantly from the tension of the pole of the flagella to cell body, will be more challenging to the cell than on a smooth surface. So that's our hypothesis. we think by creating more set area to attach, maybe we'll create a more stressful surface for the cell to attach to. With those hypotheses, and we came up with the idea that we want to create some kind of rational guidelines for this static topography for biofilm control. Basically, we think the patterns should be 10 micron or taller, so the flagella couldn't reach all the way to the bottom between the protruding features. And then we want the area to be not too big. We take the 20 by 20 micron threshold that we learned from that protruding pattern as a threshold to calculate the area that we want to make it small enough so the bacteria doesn't want to do that, because the flagella is about 6 to 10 micron in length. If they attach in the center of the feature and the cell swing around, you will get that dimension as we observed in the first threshold. Now that also became clear to us. So if we design the surface feature to be smaller than that, we think that top of the feature would be relatively anti-fouling. And then the gap between features, we want this to be small enough so the cells don't just easily fall between the features, but also wide enough so they can't just bridge over easily. We think 2 to 5 micron could be a good dimension for that. And then we also change the geometry of this shape. So the hexagon patterns we believe is better than square because it creates more size for the same area than the square for the cell to attach. And pillars, circular shape could also be good. And by doing this and testing different designs, we found one feature with 15 micron across, a hexagonal shape, 10 micron tall, and 2 micron across. You can reduce the fouling by 90% compared to smooth silicon surfaces. So it's a proof of concept that by engineering static topography based on what we learned from bacterial surface interaction, it's possible to reduce biofouling that way. Now, to do more about this, we also want to engineer some more standard testing so we can have more repeatable results and screen hopefully larger materials better library. So last year we published work based on this high throughput approach. Essentially, we take the format of 96 well plates. We can fix the biomaterials at the bottom of the well, and then we can apply a wash using a plate washer, and then to study bacterial attachment in those recessive wells. We did this with some colleagues in FDA, published this method. Now we're trying to use that to understand more about rice implant-related infections. So there's to show some example up to this point about static topographies. They have the advantage that we can do some rational design. They can show some promising result for biofilm prevention. But they are not solving the problems yet because the biofilm prevention we showed there's still about 10%. That's actually a lot of cells when you have microbial infection. So we need to do something more than that, especially if you can remove the biofilms after they're formed. So then from this, we think about back to the how bacteria read the map concept. If the map is representing like a city, cities, and then microbes build biofilm clusters as cities, then the earthquake can be a powerful way, right? Bad for us, but for bacteria though, that may be a way to shake this substrate to remove biofilms. And the way we think about that is to apply the so-called dynamic topography, essentially make the surface to move. And their materials out there called shape memory polymers has been around for a while. We're excited to find that has not been used for biofilms at that time when we did this work. So we applied that to this study. The idea was to, these polymers have different, it's a copolymer, so it has different transition temperatures. You can program that. So what you can do is you can make a permanent shape, and then you lower the temperature to fix this, and then you increase the temperature to above certain TG, but not everything. So you can then change the configuration of the surface and then cool this down to go to a temperate shape. And then when you deploy this for certain conditions, you can trigger the shape to go back to the permanent shape, essentially to trigger that shape change. During that time, you can have some dramatic movement of the surface that can be used for many different applications. In this case, we're interested in biofilm control. So as shown in cartoon here, we have this patterns we engineer, and then we can stretch this material as a temporary shape on the left, and then we can cause the recover to the permanent shape on the right. So that will be the shape change to trigger biofilm removal. So I will skip this movie here, just go to the, oh, actually it's showing, maybe we can watch that. I was a little concerned about if my pointer wouldn't allow me to show that. Just look at this one on the right. After transition happens, you can see the surface start to slide to the left. And then this movement will cause the biofilm to detach. You can see one of them showing here, this class as example, is rolling out of the surface because the substrate is sliding. So this is a quite effective way of removing cells physically. There's no antimicrobial applied here. Just by physical forces, we can remove cells. So if you do that, after this removal, we can take the surface just by simple rinsing. You can see it becomes quite clear after that. You can reach about 3 logs of removal, quite a lot more than static control that we show in the prevention itself. And then this, the other advantage of this is because it's a physical force, so it can be applied to many different species. It's not like antibiotics to be used for certain species or strains. This one has no limitation in that regard. But it has synergy with antibiotics because once you move the cells from the surface, they can be more sensitive to antibiotics because they're no longer in that protected structure. And as individual cells, especially if they move around, they become more active. Then that also addresses the dormancy issue when you have those surface attached structures and dormant persistent cells. So we found they're somewhere between 2 to 3 logs more sensitive to the same antibiotics compared to treatment of the biofilms on the surface itself. Along with that, we also found when the cells were kicked out of the biofilms, their ATP level increased, and the cells start to regain some of this growth activities. And that explains why they're more sensitive to this killing by conventional antimicrobials. So the materials I show you show some promise, but it only goes shape memory change once. So it can't go back and forth. For devices, we want to see if you can have repeated control. And actually, this kind of materials exists, basically reversible shape memory polymers is already in literature. As a proof of concept study, we took one system, we validated this can go different cycles. And as you apply this to biofilm, you can see every cycle you reduce some biofilms and you can realkylate and realkylate and go back after cycles. You can reduce this up to about 2 logs. So it proves that if you can have this shape memory polymer go back and forth, you can gradually remove biofilms through the cycles. So overall, these 2 studies, as I show here, they prove the concept that if you change the shape of the bottom of or substrate of biofilm formation, you can remove established biofilm cells and sensitize those cells to antibiotics. And then they're also somewhat programmable, or at least they're on-demand control. You can control when you want to trigger the shape change, but they are not fully programmable. And even if you can reduce this biofilm with recycling of this reversible shape recovery polymers, they will not go forever. They will have breaking point after a number of cycles. So we were more interested in finding, is there something more that we can do about this, which we hopefully to do something more we call active topography. This is inspired essentially by ourselves. We have done a lot of biomimetic studies, mimetic studies of different materials inspired by nature. But if you want to control the problems to help ourselves, maybe human are the ones we should also mimic. This is inspired by the cilia on the epithelial cell surface, as we have every day. In terms of the mass, we actually breathe in more air than food that we eat. So that sounds scary, but there's a lot of air we breathe in. They contain microbes. The reason that we can remain healthy is because we have a mucin layer across the epithelial cells when we breathe in, when they're containing our tissues. And this mucin will trap the microbes, viruses or bacteria or fungi within those mucins. And this cilia then will beat and move this mucin up as we cough out of these microbes in sputum. So if we can learn how to engineer the materials to do something like this, we would be able to have a more active surface to prevent microbial colonization. So can we do that? We tested the idea using this artificial cilia. Essentially, they're micron size polymer pillars that we can put supermagnetic nanoparticles on the tip of this using lithography, essentially microfabrication. So then we can drive these pillars to bend if we can apply a magnetic field. And then if you remove the field, then they will stand up. So if you can repeat this, then you can have this pillar to beat, right? Just like our cilia cell have this epithelial cells as cilia to beat on the surface. Using gigantic magnets certainly wouldn't be good, but the good news is that we can then consult this physics using electric current to trigger the magnetic field by going through the coil. So if you have a coil to run, and then you can have magnetic field to be generated, you stop, you can control. So use electric control that's actually very precise and highly tunable. So if you can engineer the surface into a tubular structure, then you can then apply this to the medical devices, for example, catheters or shunts that have those kind of infections to be prevented. Now, we also think if we can generate the force strong enough, we may be even able to remove the established biofilms. So we tested those concepts in a paper published a couple of years ago. So here it shows the evidence that we do have supermagnetic nanoparticles. We can form those pillars. Using SEM-EDS, we show we have aggregates of the nanoparticles at the tip, and then they do bend. And my colleague Teng Zhang at Syracuse in mechanical and aerospace engineering department, he did simulation to show the stress we can generate from the beating of the pillars. The test we have done was to take the flat control, we put bacteria down on this, or we can put the pillars, we call statics, when we don't turn on the electric current, so they just stay there, there's nothing moving. Or we can have constant beating of this, so you can prevent biofilm formation. And then if you do have biofilm formed, you can turn this to a stronger force by increasing the magnetic field strength. And then you can have stronger force to beat them out. And then where you can do a combination. So we can see there is a added benefits by using both prevention and removal. So this shows the data of UPAC, which is uropathogenic E. coli that causes UTI. Similar results were found for Pseudomonas aeruginosa. We can see pretty effective prevention and removal. And Staph aureus, so you can go faster, same idea. You can reduce biofilm formation and remove the mature biofilms. And just like I showed for the shape memory polymers, when the cells are removed from the surfaces, they're also more sensitive to antibiotics. I think this is a little busy bars, maybe just compare this to once they're removed, they're more sensitized to antibiotics. This is by close to 2 logs increase in killing. We also did some in vitro tests to show they're safe to human cells, and then effective against at the condition effective against biofilms. We did a in vitro prototype catheter, just put the pillars on the wall. We show that flat control or static pillars, the little section can be clogged by in just 3 to 5 days by UPAC biofilms. But if you have the pillars beating, this can remain clear in 30 days. So we have just finished a prototype. We're just about to start some animal tests to show. But also I want to share just in vitro at least, we saw consistent with the imaging results we saw, we also observed a significant reduction of bacterial load. So it's interesting to know that to prevent infection where the blockage of the catheter, we don't really have to go down to 0 for UTI control, but you want to have significant reduction. So this also help, I think, in the future to consider what is colonization, what is true infection. That will also provide some insights for the control and the medical device design. Moving forward, we're testing by conjugating mucin to the surface. We have done that recently. We show there's additional benefit by coating the surface with mucins to be more human-like. We also know there's zero-ionic ions, zero-ionic polymers that can be antifouling too that we can apply and integrate to this. Now I'm going to switch the gear a little bit. So there's after the biomaterials part, I want to use maybe 2-3 minutes to talk a little bit on the drug discovery side. So we can change the materials design as I show you as a possible way to reduce biofilm formation and remove this biofilms. As we're also interested in finding some new ways to discover antimicrobials that can control those dormant cells, which are highly resistant, tolerant to antimicrobials. So the concept about this is bacteria, for many reasons. This is a pretty hard area to study, and long story short, bacteria can go through phenotypic changes and go into this kind of dormancy. As shown here in the cartoon, the blue cells, there may be a small number of cells in the population become dormant. So if you apply antibiotics to this, you can kill out the normal ones in the yellow color. This will stay. And some of the normal cells may be triggered to form those dormant cells as well. That's known. So essentially, if you choose a wrong antibiotics, especially, we may end up with a smaller population than you begin with, but you may increase the population of those dormant cells. If they somehow escape the immune response by macrophages and neutrophils and other cells, they may regain this population after you start treatment to form similar composition as you begin with. So this goes back to the persistent problem as we talk about. Conventional antibiotics don't kill this. If you remember, by the discovery mechanism, we look for the drugs that can have inhibition of growth. If they're not growing, it's not the target. So the vast majority of antibiotics don't have any activities on the persistent cells. And we're thinking, can we do something different? One of the hypotheses we have is if the cells are active, they have all these components working in the cell. One of the major mechanisms of resistance is efflux. So they use ATP or other energy sources to pump the drug out of the cells. That's one of the major resistance mechanisms. We think when we go to the dormant cells, now when the cells are basically sleeping, this efflux will be reduced. Now, what about the drug influx they get into cells? Some of the drugs go through poring. This is essentially a channel allow the cell to go, the drug to go through. But because of dormancy, there's a change in the membrane potential. The decrease of membrane potential will also be less favorable for this drug to go in. So that will also be reduced. But if the cell, if the drug can go through the lipid layer by diffusion-driven penetration, we think these drugs will still be able to get in. Now, if they can sneak in the dormant cells, if the target is still available, then we'll be able to bind to the target. And then if we remove the extracellular drug, the cells will be able to wake up and try to go back to normal cells again. During that process, two things can happen. One is this drug, if it doesn't bind to target tightly, it will diffuse out. Essentially, this drug will, this dormant cells will come back to be a normal cell. But if the drug binds to the target strongly, this killing then will happen now because cells are waking up. Now the target will become effective again. So the killing can potentially happen if the diffusion is slow enough, or if the drug can bind to this really tightly, almost like covalent mechanism, they don't even diffuse out. That would be even better. So that's kind of the hypothesis we put in this to look for drugs that can go through basically empathetic drugs that can penetrate the membrane, lipid membrane through diffusion. They have strong binding target in the cells, and then this killing can happen during the wake up. So it's kind of hypothesis. We look for drugs for that kind of features and do some testing. Like I expected, most of the drugs wouldn't work because they were not selected for this. But we were, I think, fortunate to find one existing drug, is minocycline, that actually does this. It's not effective against normal cells because the efflux of minocycline is well known in E. coli. It got pumped out. But we found when we have persistent cells isolated, we show a dose-dependent killing of them up to 70%, showing the light bars. This is a dose-dependent killing test. But the dark bars showing the normal cells, they don't respond to this. So we're encouraged by that, and then wanted to know if there's anything to do better. Interestingly, there was a derivative of minocycline called eravacycline, approved by US FDA in 2018. This drug has additional group that can bind to the 30S subunit of ribosome more strongly than the minocycline. And then we found this one actually kills the persistent cells much more effectively than the minocycline as well. So remember, I just mentioned that minocycline can kill up to 70%. This one can kill up to 3 logs, 99.9%. And we found it accumulates more in the persistent cell than normal cells. And then we also tested, you put the drug in, you incubate, and then you remove the extracellular drugs by washing and put it back in the culture medium to regrow. The control one will grow back because normal cells wake up. And then the treated ones, there's no change in this culture turbidity. If you follow the live cells in that population, it actually decreased. So that's consistent with the killing we observed, showing the mechanism that the killing happens during the wake up. I think I'm going to skip a couple slides and wrap up. So quickly, we did some additional tests to show this can work on other species, and we did a short clustering analysis, and we found some new drugs that can also kill persisters. Through this quick test, we found 4 out of 10 drugs we tested can kill. So it's pretty high yield there to show this mechanism where the principle has some promise there. So I'll skip this, and to just conclude, I hope I demonstrated that the biofilm and persistent infections are important problem to solve because they're really contributing to the persistent infections that we see in many occasions. In addition to what I have discussed, the dormancy also contribute to, for example, Lyme disease and latent TB and many other cases. So that needs some kind of paradigm shift to think about how can we address this in different ways. We think material design can help, and new ways of doing drug discovery can also help. And there's synergy that we can create by using different approaches. Finally, I want to just thank all my funding agencies. The ones that supported the data I presented today are listed here. Also my collaborators and some previous lab members who contribute to the work that I talked today. So with that, I think I will stop here and be happy to take any questions.
Thank you for the fantastic talk, Professor Ran. Several questions came in. One of them is, what are the pros and cons of topographical cues in resistant bacteria, and what is their effect?
Oh, sorry, just to clarify the question, was I asking?
What is the advantages and disadvantages of topographical cues?
Yeah, that's a good question. So using topography, I think this is more of a physical way of doing this. I think when it comes to resistance, I think it does carry some advantages. The way I think, in my view, about resistant problem is when you have an infection, you give antibiotics. Essentially, we're giving the bacteria 2 options: death, which is cure, or survival, which means resistance. The way we use physical force is basically to remove them from the surface. We're not killing them. If they go back to planktonic stage where they're swimming around, how they came from, I think we're giving them essentially the 3rd option. Especially if they're living in the human body, they can go back to nature. There's no killing to happen. So we think the selection pressure will be much lower. That's kind of our speculation or hypothesis regarding this. I don't know if that answered the question.
Yeah, it's pretty answered it. Second question is regarding dynamic topography. Can you translate this to the clinic? Are there any difficulties?
Yeah, so this is a good question. I think this will be some topical modification. I can say I can't say everything because there's some potential IP things there. But I think it can be translated into certain devices that we can change the surface features, especially softer materials. We can't do this for example, knee replacement. That's not the place, but some other soft ones possible.
Thank you. As you know, we do a lot of hydrogel biomaterials. The question is, what design strategies would you recommend to make hydrogels antibacterial?
Oh, that's a good question. I think so. Certainly, topography would matter, but I think the stiffness also matters a lot when it comes to hydrogel, especially those very soft materials. Um, I think that part, there's actually a lot of literature in there. We happen to be fortunate to be one of the early ones to look at this. I remember, which I didn't talk today, just by taking the PDMS, for example, you change the Young's modulus, you can see a major difference in there. But later studies shown that in addition to stiffness, there are also elasticity and other things that can happen. And if you have hydrogel, I think the pore size and many other things could also matter. So it's a quite complex picture.
Thank you. Regarding the bacterial adhesion mechanisms you showed, is it universal or does it apply to all bacteria species? Can the adhesion mechanism change depending on specific species? Are they reversible or irreversible stage, or at reversible or irreversible stage?
Yeah, well, that's an excellent question. Yeah, it does vary. Bacteria, when they come to surface, they use many different mechanisms. The ones I show are flagellated cells. Essentially, they have flagella to move. E. coli has that multiple flagella. Pseudomonas has one pole flagella. And when they use the flagella to get the initial contact, they may also engage pili, which has shorter structures on the surface to attach. And then there are also bacteria such as Staph aureus or some other gram-positive bacteria. They don't have flagella. They just roll on the surface. So the mechanisms are indeed different. I think when you come to that kind of topography control, we do have to take it into account about the species we're seeing. If you go dynamic or active, I think it would matter less because we have more control of the surface. If it's static, I think we have to consider more about what species we're interacting because they do have quite different mechanisms for that.
Thank you. Have you looked at the intrinsic antibacterial property of nanoparticles you have used to create active surface topography?
Oh, the nanoparticles? Yeah, we didn't. In fact, we use this iron oxide. They are not antimicrobial, but I think if you further decorate this with other things, you could have physical, chemical combined. Is that the question, I guess?
Yes. You want me to repeat the question or what?
Oh, I think I sort of cannot answer that way. Just like the particles we use didn't have antimicrobial activities.
Okay.
But it could be engineered with others. For us, we just want to have the magnetic response.
Thank you. How do you get the dimensions in the active topography, the design principles?
Oh, yeah, that was mostly we try to get try to get something close to human cilia, not exactly the same dimension. We were more interested in the spacing between them. But the pillar we used, they're all 2 micron across, 10 micron tall in the data I presented. Currently, we're doing some variations. I think human cilia is in the same range, maybe a little longer than that.
Thank you. For the dynamic topography, have you tested your surface topography in vivo? If so, how do you control the temperature change in vivo?
Oh, good question. We haven't. I think I have a colleague who has done some other work. I can't speak for their lab, but I think they're moving to that direction. In this particular one we did was between room temperature and 40. So if you do that, you have to consider again, this one, sorry, I can't say too much, but there are certain devices you may consider, or maybe topical dressing you can consider that may give you that. But I want to add though, the trigger of what we did with temperature was more of a proof of concept. The shape memory change can be triggered by many others. I would say temperature is not the most friendly one for in vivo applications, but others are. For example, pH, electric current, magnetic, or even enzymatic triggers. Those can be engineered for that. I think if you think about in vivo applications, you want to consider that way.
Thank you. For active topography, the SEM picture showed that your pattern surface is in micrometer scale. How can it be scaled to the actual implant size? What technology is available in market to make such a pattern?
Yeah, they are made with soft lithography. So essentially, if you make a wafer large enough, you can make it bigger. But you can also consider, we haven't done that, but I think in certain cases, you can consider 3D printing, for example. So there are a lot of modern advanced manufacturing technology can be applied if you really go down for translation.
Thank you. A final question, does biofilm thickness matter besides biomass or surface coverage?
Yes, we do mostly, so there are two ways we do. One is we do biomass, which is the 3D imaging to include everything in that structure. Or we do CFU, which to count number of live bacteria in that structure. I think if you only look at 2D coverage, it wouldn't tell the whole picture because they usually have multiple layers, especially the ones we found with polymer as a substrate. They can form quite thick biofilms.
Thank you. If we looked into the hydrophilicity or hydrophobicity of surfaces to control bacteria attachment?
Yeah, that's a good question. We look at a little bit. Most of the thing we did, like the dynamic topography or active topography you showed today, they're just PDMS. They're rather hydrophobic. If we modify them, such as we mentioned, like a mucin or zivotonic polymers, they become quite hydrophilic. We actually want to make them to be super hydrophilic. So the field knows that if you make them super hydrophilic or super hydrophobic, either way, you can reduce biofouling. So you want to go that direction if you want to use hydrophobicity as a control. But there are also somewhere in between if you integrate other factors, I believe.
No, that's all the questions I have. Thank you for your time and the fantastic talk, Professor Ren.
Oh, great. Pleasure. I try to speak a little bit fast. I'm watching time. Hope I answered those. If not, feel free to contact me and I'll be happy to connect after this talk.
Thank you.
All right, I'll stop sharing.